Hello everyone, I am Dr. Kavita Gajbhie, Assistant Professor in Pune College of Pharmacy, Pune. So today I am going to deliver um, uh, a talk on the topic differential staining, or it is also known as gram staining process. So first of all, it is very necessary to understand what is the gram stain. Gram stain in late 18th, Christine Gram observed that some genera of bacteria retained a diiodine complex when rinsed with alcohol, while other genera were easily decolorized with alcohol and could be then visualized by a contrasting counter stain. So, on the name of Christine Gram, who was the founder or who was the discoverer of this gram staining process, that name of process was happened gram staining. This staining process are defined two bacterial groups: those which retain the primary dyes, which is known as the positive by gram method or gram positive method, and those which are easily decolorized. So it is known as negative by gram method or negative gram negative staining. This is the starting point for bacterial identification process. So what is the difference in the dye? Because the difference in the dye retention is dependent on such physical property of the bacteria. For example, we can say the thickness, density, porosity, and integrity of the bacterial cell wall, as well as its chemical composition. So gram-positive bacteria have thick, dense, relatively non-porous wall, while gram-negative bacteria have thin walls surrounded by lipid-rich membranes. Some non-bacterial organisms with thick cell wall, for example, few yeast molds also stain gram-positive, and bacteria which have lost their integrity through aging or physical chemical damage deterioration may also stain by the gram-negative staining. So in this diagram, you can see the composition of a bacterial cell wall. It depends on the thickness of lipoticoic acid present over the cell wall formation and peptidoglycan plus teichoic acid combination. So in positive, gram-positive bacteria, you can see there is a thick composition or thick cell wall occurs as compared to the gram-negative bacteria. Due to this physiological difference or difference in the integrity or composition of lipoic acid, lipoticoic acid, polypeptidoglycan present in the cell wall, it shows different kind of staining, positive and negative, because it shows a kind of interaction with the basic and negative dyes. So gram staining procedure required some kind of primary stain, for example, crystal violet, some type of mordant, for example, iodine solution, few decolorizer, for example, ethanol, and secondary stain, which is also known as counter stain, for example, safranin, and obviously water for washing or for cleaning the excessive amount of stain over the slide. So what is actually the gram staining procedure? Here first the bacteria are stained with the basic dye, for example crystal violet or primary, as we have already discussed in previous slide, and both gram positive and gram negative bacteria become directly stained by using this primary stain and appear purple. The bacteria are then treated with gram iodine solution, which is the mordant, and this allows the stain to be retained better by forming an insoluble crystal violet iodine complex. So this complex, in case of bacteria, can be able to produce in case of gram negative or either in gram positive. So both gram-positive negative bacteria remain purple in initial stage, and after using the decolorizing agent, for example, ethyl alcohol and acetone and ethanol, this is, we can say, the differential state, because after decolorizing, using of decolorizing agent, only gram-positive bacteria retain the crystal violet iodine complex, while gram-negative bacteria are decolorized. Finally, the secondary stain, For example, safranin as a counter stain also we can say. We can use in the last stage, and since gram-positive bacteria are already stained purple, they are not affected by the counter stain. But gram-negative bacteria, they are decolorized previously by using decolorizing agent. So it become directly stained by the safranin as a secondary stain. Thus, gram-positive appear purple and gram-negative appear pink. So you can observe the identification or stepwise addition of different kind of stain in this slide. Here you can see stain purple initially remain purple after using the iodine, and after using the decolorizing agent, gram-positive bacteria remain purple but gram-negative decolorized, and here also at the last stage, gram-positive still remain purple but gram-negative colors pink or orangish in appearance. Yes, positive and negative you can observe. Now, what is the meaning of acid-fast staining? So here, the cell wall of acid-fast bacteria are quite different as compared to positive and negative. Here you can see the cytoplasmic membrane, a thick cytoplasmic membrane is present, and then moderate kind of peptidoglycan layer is present, and over there some kind of lipo arabinomannan and mycolic acid. Mycolic acid is the speciality of acid-fast bacteria or acid-fast cell wall. Due to the presence of this mycolic acid, it shows a kind of acid-fast staining or use specific kind of stain. So free mycolic acids and peptidoglycans are also present over the surface, and arabinogalactan are present. You can see the component or composition of different kind of lipids, different kind of that peptidoglycan in case of acid-fast bacterial cell wall. So once stained, in case of acid-fast staining, once stained, the acid-fast bacterial cell resists the decolorization with acidified organic solvent, and we can say therefore that's why we can say it is acid fast. And for example, the mycobacteria genus, that bacteria generally shows this kind of acid fast staining. It depends on their lipid rich cell wall, which are relatively impermeable to various basic dye unless the dyes are combined with phenol. And the exact method by which the stain is retained is nuclear, but it is though thought that some of the stain become trapped within the cell and some forms a complex with the mycolic acid. So after formation of complex, this supported by the finding that shortened chain mycolic acid or mycobacterial cell with disrupted cell wall stain weakly acid fast. And for example, we can say nocardia is also one of the bacterial species, acid-fast bacterial species. You can see here some kind of representation of acid-fast staining. So here you can see some reddish acid-fast bacilli among the blue normal flora and white blood cells in the sputum that are not acid-fast. So some kind of differentiation you can easily see in this slide. And now, after that, I am coming to the point, what is the spatial staining? Because see, staining is positive staining, negative staining, both are comes under the category of that simple staining process. Or we positive, we can say the direct, negative, we can say the indirect staining process. And after that, if we see some another kind of classification, so here we have already taken the differential staining, after that the acid-fast staining. Now I am taking the spatial staining process. So spatial staining process, here some specific kind of component that we can easily stain by this process. So capsules, flagella, granules, that we can easily identify. So if we are taking that capsule staining, so what is actually the procedure that we will discuss? Capsule staining is diagnostically useful since it is a virulent factor. For example, in case of pneumococci. So bacterial capsules are non-ionic, so neither acidic or nor basic in nature, will adhere to the surface. And capsules are demonstrated either by negative staining, nigrosine or Indian ink as a dye, or by special staining, for example, His method or Anthony's method. These method named on the name of the discoverer. This method is what? The capsule is non-ionic in nature, so it doesn't get stained by an acidic stain, but a basic stain such as crystal violet stain the cell wall as well as the capsule. So it can be easily stained by the crystal violet. So this is followed by treatment with the hypertonic solution, 20% copper sulfate solution with reverse dual role of both decolorizing agent and counter stain. And copper sulfate solution being hypertonic cause diffusion of stain toward outer surface of the cell. After drying of the slide, the stain which is not passed from the capsular layer during diffusion retain in the capsular layer. Copper sulfate then decolorize the capsule, and capsule appear as a faint blue hollow around a purple cell. So this is a kind of representation stepwise. Prepare a smear, air dry, doesn't heat fix. Flood the smear with the crystal violet for 4-5 minutes and rinse the slide with 20% of copper sulfate solution, and again air dry the slide and observe under oil immersion lens. Now I am taking one more example of spatial staining, which is endospore staining process. So here endospore staining is what? Spores are normally impervious to stain. Under the light microscope, endospore have a high light refractivity uh, indicative of the high protein content and can be stained by the modified Gill-Nelson method using 0.25 to 0.5% sulfuric acid as decolorizing agent. Bartholomew Mitvar method, Schaefer, Fulton stain method, lots of methods are there. So yes, these methods are also based on the identification of a specific kind of stain on the slide. So here you can see green endospore within the pink bacilli you can observe in this slide. So all these are the different kind of staining techniques that we have discussed. So all these are the reference for the staining process. Thank you so much.